Flowering plants, also known as angiosperms, dominate our entire planet because of a highly specialized reproductive organ, the flower. Morphologically, the flower is a modified shoot. In a standard plant stem, leaves are separated by sections called internodes, but in a flower, these internodes are highly condensed. As the internodes compress, the standard vegetative leaves are radically specialized into reproductive appendages. To understand this blueprint, we are going to build a perfect flower from the stem up. Mapping this structural organization reveals the precise mechanical requirements for plant sexual reproduction. The foundational axis of the flower starts with the pedicel, or stalk. The tip of the stalk swells to form a central base called the receptacle, or thalamus. 4 concentric rings of specialized leaves, called whorls, build systematically upon this thalamus foundation. This division creates a strict dichotomy. The flower is split into an outer ring of sterile accessory whorls and an inner core of fertile essential whorls. The sterile whorls do not produce gametes. They exist to provide physical protection and attract pollinators. This structural split separates the superficial packaging of the flower from the actual genetic engine inside. The first, outermost whorl is the calyx. It is composed of individual green units called sepals. The calyx provides mechanical protection for the internal organs while the flower is in its vulnerable bud stage. The second whorl is the corolla, which is composed of petals. These petals function as visual and olfactory attractants, using color and scent to draw in biotic vectors like insects and birds. In species where the calyx and corolla are visually indistinguishable, they are collectively called the perianth, and the individual units are known as tepals. While petals are often the most conspicuous part of the flower, they exist solely to facilitate the survival and cross-pollination of the hidden reproductive organs. Removing the sterile whorls exposes the fertile core. This third ring is the Androecium. The Androecium is the male reproductive component of the flower. Its individual unit is the stamen, also called a microsporophyll, a specialized leaf that produces microspores, which become pollen. As we isolate a stamen, we see it consists of a slender supportive stalk called the filament. At the top of the filament sits the anther, the active site where pollen is produced. The stamen is an elevated dispersal platform. The plant calibrates the length of the filament to place the anther in the optimal position for pollen transfer. A transverse cross-section slice reveals how the anther is organized internally to manufacture pollen. Most angiosperm anthers are bilobed, meaning the structure is divided into 2 distinct halves. These lobes are diathecous, meaning each lobe contains 2 distinct chambers called theca. This organization creates a tetrasporangiate structure, housing 4 total microsporangia, or pollen sacs, in the corners. The microsporangium wall is built from 4 layers. The innermost layer is the tapetum. The tapetum provides multinucleated nourishment to the developing pollen. It also secretes sporopolanin, a highly resistant polymer that forms the protective outer wall of the pollen grain. The internal layout of the anther functions as a highly stratified biological factory, essential for male gamete survival. At the exact center of the flower is the fourth whorl, the gynoecium. The gynoecium is the female reproductive organ, made of units called pistils or carpels. These are megasporophylls, specialized leaf-like structures that produce megaspores. A carpel consists of 3 zones. At the top is the stigma, the receptive landing pad for pollen. The stigma is connected to the base by the style, an elongated, slender tube. At the base is the ovary, a swollen chamber where the female gametes are stored. A gynoecium may have one carpel, known as monocarpellary, or multiple. When multiple carpels are fused together, the structure is described as symcarpus. This three-part design acts as a specialized biological funnel, capturing pollen at the stigma and routing it securely to the ovary. Looking inside the ovary reveals the internal anatomy where the actual gamete resides. The internal cavity of the ovary is the locule. Inside the locule is the placenta, a cushion of tissue where reproductive units are attached. A stalk called the funiculus attaches the female reproductive unit to the placenta. The structure at the end of the funiculus is the ovule, or Megasporogenesis. The ovule is the precursor to the seed, housing the embryo sac and the female egg cell. This deep internal placement shields the fragile egg cell from environmental hazards until the moment of fertilization. Reassembling the flower allows us to see how these four whorls work in concert. A pollination event occurs when an anther releases a mature pollen grain. Once a pollen grain lands on the stigma, it sprouts a pollen tube. This tube grows through the style tissue to reach the ovary and enter the ovule. The static architecture of the flower exists solely to facilitate this dynamic pathway for genetic transfer. This matrix summarizes the blueprint. A complete flower requires all four whorls working in absolute coordination. The outer whorls protect and visibly attract, while the fertile whorls produce and house the gametes. This physical coordination allows the pollen tube to deliver two male gametes to the embryo sac. This triggers double fertilization, where one sperm creates the embryo and the other creates the nutrient-rich endosperm. Understanding these anatomical classifications provides the foundational blueprint required for advanced biological study. The flower is a mechanical marvel that transforms a simple vegetative stem into a perfectly calibrated reproductive machine.